It is a great pleasure to introduce Sanju Sinha, who's an assistant professor in Sanford Burnham Prebys. He's leading a lab focused on developing better understanding, diagnostic, and treating cancer with the help of AI and machine learning methods. He is trained as both a bioengineer and a computer scientist. He has done his PhD from the National Cancer Institutes. He has accolades which include awards like Emerging Leaders of Computational Oncology, given by Memorial Sloan Kettering, Best PhD Student at NCI in 2021, and Best Postdoc at NCI in 2023. And we found Sanju through some of the medical scientific peer-reviewed articles that have published on AI and cancer, and it's such a pleasure to have him here. And I think he's also excited to meet all of you and share some of his insights on the world of AI. So go ahead.
Thank you, Sarah. This is very nice. Before I start, I really wanted to thank everyone here for their time, as well as what an honor it is to come and present here. I've been, of course, very fortunate to be present at multiple places, having very reputed scientists, but for the first time, I'm actually presenting my science to individuals who are facing the disease so closely at such scale. So it's really the highest honor that I have, and I'm very happy to tell you what we are working on. And while we hear a lot of hype about AI, I wanted to show you behind the scenes of what are the challenges that we are facing, what is the excitement, and how the field would look like in a few years' timeline, and what are the benefits that you will receive from this hard work. So today what I wanted to talk about is the next generation of treatments and diagnosis that AI is enabling. So what I would try to cover today is that where AI is right now, what is the promise of it, what is the reality of it, what are the bottlenecks that researchers like me, patients like you are facing in this field, and what are those specific problems that AI can help solve. Then I would briefly tell you about a few tools and discoveries that my lab has developed in the last few years. Followed by that, what are a few take-home messages that you can take. Starting with some specifics beyond the hype of AI that we hear about it, is that in 2020, is there a pointer? Yeah. In 2020, for the first time, this tool called AlphaFold 2 was able to solve this problem of protein structure. And why is this important? Is that while we hear a lot about DNAs, RNAs in cancer, we actually don't hear too much about proteins. It's simply the reason is we don't know how to deal with them. We don't hear about proteins. It's because they are far more complex to deal with. They look really complicated, like here in the right, instead of just sequences. And AlphaFold 2 was able to make predictions that how does this protein's structures look like from their sequences directly. And how it can help you if you have a mutation, let's say exon 11 mutation, it used to take us previously years, more than 5 years on an average, to find how does a protein structure really looks like, to find even a drug for it. And this was one of the major challenges in the field of drug discovery. But for the first time now, if a new patient comes, and if there is a new mutation, we can predict how does the protein may look like and start on the go what kind of drugs we may want to develop here. And this, like at a very basic science level, how AI is starting at the fundamental. This was actually the method of the year for like 2020, really advancing over. So just to give you an idea, in 1950, we identified what is a DNA, right? And it took us 70 years from DNA to actually try to predict how does a protein may look like. So that's how much of an advancement this tool has been able to make. So the next one is FDA approval for cancer pathology, where if you have a pathology image, AI tool can automatically identify and diagnose what grade of like prostate cancer it would be. Okay, start from there. So this company called Page got this first FDA approval. Another one that's really, I'm sure many of you might be hearing about is called these big models, language models, right? So these big language models are exceptional in answering medical exam questions, almost as high as if you have 60% grade that you need to do to actually pass this medical exams, they reach as high as 86.5%, okay? Suggesting that now we are reaching a point where if you have medical information in diagnosis, these language models could be greatly informative and to the physicians. However, the one that really is very important is this model called AIM. And when we actually put in place and did testing with patients, something that we learned is that these models have a higher level of empathy towards the patient compared to the doctor. This is, as a first knee-jerk reaction, we are like, how is this possible? Like, how can it have more level of empathy than doctors? But if you think about it more carefully, it is actually really makes sense because doctors also are overburdened, have so much like load overall. So suggesting that these models could assist them to decrease that load is really exciting direction that it could go. So we can imagine a world where indeed we would have this model as an assistant to doctors can focus on more important problems, and these models can help them solve the information problem, the load that they are getting right now. However, while these really promising things are occurring, at the same time, so many fields are investing hundreds of millions of dollars towards AI. But there is also a reality check of AI, is that the whole pharma field is trying to use this to try to develop new drugs. But when there was an independent testing, they actually showed that in the last 10 or so years, there is no help that AI has been able to do in drug discovery. It has not been able to decrease the cost of it. It has not been able to make fast. So just to show you this data is that the amount of time it takes for a drug to like develop, it's increasing only, starting from like almost last 70 years, suggesting that AI is not really helping it to decrease. On the other hand, this blue plot shows the amount of money that it takes to create a chip. That is the Moore's law, right? That how it keeps decreasing. The computation is becoming more and more cheaper, but drugs are becoming more and more expensive. And AI is not really helping that. So while there is a lot of excitement, there is also a reality check behind it. Now, understanding both aspects of it, let's understand a little bit more that what are the key bottlenecks in cancer research specifically that AI can help resolve. And I want to start with the first one, which is the most, in my opinion, could be directly applicable to patients that are right now trying to find new therapies, is matching of patients and clinical trials. And I was very briefly explaining, actually, during the roundtable, that I like to say, from an objective perspective, this is almost like a dating problem, okay? That patients are trying to find clinical trials with a lot of information that they have with themselves, okay? Some of them, even they don't know, right? And clinical trials are trying to find and recruit patients, as for example, one of the few sessions before, they were talking about a clinical trial and recruitment as well. But even they don't know all the information that it seems that this is the case, but it's very messy. So a matchmaking of these two and using via AI language models can really help advance this. So there are very exciting advancements occurring at this stage. So I can assure you that this would be something look very different in a few years timeline that you would get benefit from, and hopefully as the next generation of patients, they could get the most benefit as well as you. And many more applications that are there. For example, early detection of the disease that could be personalized treatment with so much information that one learns from patient. We actually don't know yet how to utilize it in the best fashion. While we argue that, oh, we are really putting forward these advanced models, at the same time, we don't know how can we really best utilize the information that we are learning from the patient. So we are also like improving quite a bit and have a huge space to improve. Faster diagnosis and new treatments as well. So I want to give you an example of one of each one of them that I am developing in my lab, okay? So the first one, I like to call it that monitoring tumors one cell at a time. This is the discovery that my lab did, and based on which actually Sarah invited us here. And why do we care? Why do we want to monitor cells one cell at a time? while you have heard in the last 2 days a lot about tumor heterogeneity, right? Tumors are very heterogeneous. If we give them a therapy, they potentially have a resistant cells. There is also one more major issue, adaptability. Tumors are also very adaptable. If you give them a therapy, they can completely cut off the whole line of usage of that particular information or that particular, let's say, metabolites that they're using and go in a different direction and utilize different source of energy. So they're highly adaptable. And this is actually, I'll rather argue, is a major challenge than even heterogeneity. And we don't talk about it much. The reason is we are not good enough to, how can we really address it? Okay? We talk about heterogeneity because we have technologies to try to address it. But how are we addressing it? The problem that this cause of adaptability and heterogeneous is that resistance to therapy is a very common phenomena that we observe. And this really limits the benefit that patients receive. So what is the need? The need is that if we can have a very high resolution method to monitor these tumors, almost at a single cell resolution. So what the bottleneck here is that while this is great, we will try to develop a technology that is at a single cell resolution, we don't have data to really do that. So how does we try to solve it using computation? What we do is develop this really fancy method, okay? But the important thing is not this diagram, but one thing to remember, the first step is that we first learn from already existing data that is there, really, this major big models, okay? That we have a lot of data from existing patients, how can we learn from that? And then we actually refine our models using these single cell models, okay? And that's why at the end of the day, we can develop a model that could be utilized at a single cell resolution and can help us develop and monitor tumors at a single cell resolution. And what can we do with it? We can actually, let's say this is a patient, and x-axis is time, y-axis is resistance. We can monitor when the patients may get resistance very early stage. And this can help us identify how can we shape our treatments, even potentially change the treatments way ahead of the time before the resistance occurs. This is not just for one patient, we also have for multiple patients that we see this is the case. Now, what is the final output that we get? At a high resolution monitoring method for the tumors. Now, what is the next one? I like to call it that this is a Google to find drugs. Is that the current problem is that, let's say in the case of GIST, we identify a specific driver for you, right, for your mutation. However, finding a drug for that is extremely hard. As I explained, there are so many challenges that is there. So that's why there is no a connection between actually finding a new mutation and creating a new small molecule that can target it. So we really don't know how to develop that. And if you think about it, again, this is a problem with computational or people who are trying to develop drugs, that they are not good enough again, okay? So what do we want to do about it? What we want to do is to develop a model or a computational model that can generate a precise drug for a given gene and a mutation. Okay? What is the bottleneck? Why have we not been able to develop that? We have not been able to develop that because, again, the issue is that we don't have enough good data to try to solve this problem. Okay? Now, how do we do that? We develop, again, a machine learning model to try to do that. I would not go into the detail of it, but I can tell you that there was a Nobel Prize that really helped us develop that, is that from CRISPR-Cas9 from Jennifer Doudna. And her technology is the one that allowed us to learn that, oh, can we identify drugs that are very specific? And what we did after learning this is applied it to the current set of drugs that are there, okay? And what we learned is actually quite intriguing, in my opinion, is that the current set of drugs that are there, some of them, what I wanted to show you here with this plot is that they actually not only target the mechanism that we thought it targets, but also targets other mechanisms, okay? And that's why we really see these examples of Gleevec with one example that why it was approved for one cancer type, but it is now given in another cancer type. But for the first time, instead of doing it just anecdotally, we want to do it systematically. We want to identify what are the different targets that this drug has, and let's go back and do this completely systematic, this repurposing of drugs, right? We're also, this is the first step, we believe. This is a work in progress that we are moving on, we are building on. So what is the key bottom line at the end of it? We think, firstly, there's still a work in progress. We are developing it. Second is we need a lot to do in drug development, far more specific drugs, and the current version of it, it would improve in the next few years. So what is the final one? And this is the one that I'm most excited about to tell you. I wake up in the morning for this particular one. I go to my fellows, I bother them. They hate me so much, really, because I ask them all the time about this. It's like, what is going on? Tell me. Here's the question. While the precision medicine is that for the case of just if you have a mutation, how can you match it to a therapy, right? But identifying that mutation itself is a challenge. It takes a lot of time. It's very costly. And while a considerable population in the United States can reach that level of sophisticated therapies or approach, a lot of it cannot. In underserved parts of the United States, they cannot get that, even learn the mutation is there. So what we want to do is to cut down this time as well as this cost that is there. And how can we do that? The way we think is that the challenge that it presents is that 80% of the world almost cannot afford it, and we lose very valuable time of patients. Now, why haven't we been able to solve this? The issue is that there is a bottleneck. Our current technologies are sequencing based. We take your cells, tumor biopsies, we sequence them, find what is the precise mutation. But actually, we realized when we were working on this in the last few years that there is a different way, a far more easier way, and a way that was almost created 70 years ago. We just never looked at it. The way is imaging, okay? If you have a tumor biopsy, we take pathology images of them quite commonly. This is standard of care, okay? Every hospital in the world has this method where they take a tumor biopsy and they almost take an image of it, okay? But all they do is to identify whether there is a cancer or not a cancer, okay? And a few other very simple pathology tasks. But we realized you can learn so much more from these images than just telling them what cancer type it is. We actually realized that if you put them in a really fancy machine learning models that you may have heard about, all this ChatGPT and stuff, right? But the fundamentals of it, okay? It can identify features from it that can help us predict, okay, the levels of mutation, like which mutation the patient may have. Potentially predicting whether, let's say, whether a precise driver of the cancer is there or not there, and based on that, choose therapies. So there are like 130 such mutations that are there that are FDA approved in the United States right now, and we are developing a method to try to predict it completely from the images that we believe could hopefully benefit these precision oncology methods to the underserved parts of the United States as well. What is the key bottom line? This is the imaging-based method to predict biomarkers. We are setting up this right now, the first set of even technologies that are there. Even imperfect, we are setting up with hospitals in San Diego, working with tumor boards to help them at assistance level right now to provide this information. We think that we will learn back this information and how can we best potentially implement it. I was also talking to Sarah about this, how can we implement it to make the cancer registry of GIST far more richer than it is. So these are the kind of AI models that I think we would like to collaborate with you and work together to apply to this cancer registries. How much time do I have? It's okay. So what are the final take-home messages that I have? 3 methods that I presented today. One is to monitor tumor at a very high resolution, single cell resolution. Second is a method that a world where I would like you to imagine where we can have a Google for drugs. Okay. It does not need to be IP'd or extremely a patent or secret to pharma, that they trying to make so much money out of it. But instead, if you have a particular mutation, a doctor can look up a drug that is specific enough, fits your profile, and could be given. Okay. It's a bit hard to imagine because the whole pharma industry or medicine right now is almost based on this, that, creating new small molecules. But I assure you, the methods that are developing right now behind the scenes, okay, one that I just gave you a little bit glimpse of, they would be able to make this, enable this. And it would look very different in a few years period. And finally, the method of trying to predict the biomarkers, mutation information from pathology slides directly so that it can help the cancer outcome across the United States, including the underserved parts of United States and globally. Something that I really wanted to pass you as a message is that the fields where AI has been successful is the fields where there is large and big data. And the fields that AI has a lot of hype, but actually not successful, are the fields where people claim a lot, but data is actually not there. So if next time someone tells you about AI, ask them, what is the data? Okay? And where is the data coming from? And if someone asks you a lot of money as a startup, always ask them, what is the data that you have, okay, behind that? So it is the data is the king and not the AI behind them. And that's why I think all of us as a center, I think individually could contribute here by trying to create a secure, high quality and integrated data from GIST. And I think this can enable us to try to take advantage of the advanced AIs that have been developed in the last 30 years or so period and create better therapies. With that, I would stop.
Related to that question, speaking of the data, are you leveraging, I'm thinking maybe good biobank data to be able to test your models? And from where? Thanks.
When you mean biobank data, which one?
Or like, how are you, which populations are you testing those models out on? And I'm just thinking that since you're associated with UCSD, perhaps they have a good research bank to tap into. I don't know what the diversity of that research bank is, but those are my questions. Thanks.
So in a similar way, both the answers have similar questions. The first one, which one of them we are most advanced in? Actually, the one that we started, the latest one, is the most advanced one. Okay, the one about, because we are very close to tumor boards, we are collaborating with them right now to implement it with the tumor boards at Radius Children's Hospital in San Diego. These are pediatric tumor boards, and these are pediatric cancer that are very hard to treat. So we are telling these tumor boards what is the unique information that we can provide them and how they can help them choose the therapies. I'm actually implementing this in a few different hospitals right now in talks, so we are trying to do that. So we think that this is the one that we started, the latest one, and that's why I bother my fellows too much, because I want them to really move quite fast in this direction. The answer of the question that you're referring to about Biobank, we think 100% right now that the methods that are currently in place only utilize a lot of European American data, which is primarily what cancer, most of the cancer precision medicine is based on. However, diverse data is extremely critical. We are actually working with UCSD to get Hispanic population, working with JHU to get African American population. Actually, I have like ties back from India so that I can get Indian population and a few other ones. And we are still this work in development. I would love, and I was actually talking to Sarah also, I would love to have like diverse population. And I think this could help us to develop methods that are better and find opportunities that are very unique and help individuals across the whole spectrum. So both of them are very important questions. Thank you.
I had another question. So of the three that you spoke to, would all of them be considered software as a medical device? And I'm just thinking about the FDA approval and how you're thinking through that.
Thank you. Actually, firstly, I have not thought of it till now. Okay, we are not there yet in our opinion. This could be either a medical, either a device or a software, one of them. I don't know, maybe a mixture of them as well. But at this stage, the next question that we are potentially trying to answer is the tumor board, that how can we help them? But how we could end up being, we have not thought of that, so I don't know.
Hi. So the data is king, it's not the AI point. I think that's really important. And I'm curious, when applied to medical settings, privacy and access to patient records outside of registries and things like that, how much of a barrier is that right now? I think when we talked in the roundtables earlier, thinking about someone's primary care versus their cancer treatment records may not be in the same database, or you're not looking at that when you're doing cancer research separate from all the other indicators. Are there barriers in just getting all the data together? One, because it's a big job, and two, from privacy constraints, and where are we in that timeline?
Sure. So at UCSD, what we are trying to do is to create basically a centralized repository of all the EHR data. Okay? I personally want to tell you, I think that it would be one of the most important advancement in data science AI that we would do, right? There is no AI there, and this would be the most important one, simply because of the integration of crosstalk, that how, like, in some way, we think about disease as just one disease, but not how it's holistically, like actually affecting other parts, and how diabetes and cancer is talking to each other is extremely critical. So EHR data being put together would be a huge step forward, and we are doing that right now at UC. Really, really important direction, I'm telling you.
I'm just wondering, the LifeRaf group has a patient registry where a lot of people consent to sharing their mutation and their CT scans and whether they're progression-free. When you mentioned the picture and the secure data, I'm just wondering whether a group like the LifeRaf group could request cell imaging from the pathology for GIST patients, and whether, if you were able to get that in a large volume, whether that could assist you in some way, whether it's worth the LifeRaf group asking for that, because I know there's a lot of privacy and consent issues, and it's harder for doctors to ask for that. So I just want to throw that out, that would that be something that would be useful that the LifeRaf group could do?
Thank you. Firstly, I want to say very kind of you to really say that. From my perspective, I wanted to help you, okay, to apply these models here. And you are saying that you want to help me going back. So it's really kind of you to think it that way. Yes, the answer is I have been talking to Sarah and Ian as well, that how we could make that happen, marry these technologies to LifeRaf registry to take a step forward. Thank you so much.